So good morning, everyone. Thanks everyone for joining today. So this is our second session of this essential GIBA concepts and techniques lecture series we're organizing as part of the GIBA network. Today, we're going to be focusing on the brain, specifically talking about brain processes, but also in terms of cognition and behavior. We've had our first session on Monday on the gut, was presented by John and Leslie. I felt it was quite interesting, and I think they gave both a very nice overview of the gut and the gut microbiome. So for those who are joining this for the first time, I wanted to clarify, so the mics and cameras are off just to avoid any technical issues and interferences, but I do encourage you to use the chat function to ask any questions. So we'll have time about 15-20 minutes after each of the talks for questions, so do engage through there. The lectures are being recorded. There were some questions on Monday about when they will be available, etc. So we're waiting until all three sessions are finished this week. So we'll have the next one tomorrow. And then the videos will need to be lightly edited. So the videos are quite long. We'll need to break them down into individual talks and trim them a bit. But then they'll be made available on our YouTube channel for everyone to watch. So just bear with us a bit longer. So today we'll have two speakers, so Marta Camacho and David Sanderson. We're going to start first with Marta. So Marta is a senior research fellow at the Department of Clinical Neurosciences in Cambridge with expertise in neuropsychology and longitudinal studies in Parkinson's disease. Her specific research is focused on the gut-brain axis, especially how constipation is associated with cognitive decline and faster progression in dementia. And she studies the mediating mechanisms underlying the relationship between this and Parkinson's disease. And she's going to give us a bit of an overview of brain process, neurodegeneration, and disease for this particular session. So, Marta, over to you.
So
Excellent.
thank you
very much.
Alex, for inviting me. I pondered on how to structure this talk because, of course, 40 minutes is not enough to really convey the core concepts and techniques that we use in neuroscience, and particularly clinical neuroscience. So I was just telling Alex and David that my goal for this talk today is to foster some interest in particular areas within neuroscience that people can either research further or potentially have ideas and seek experts within a certain field and establish collaborations with. And I thought, instead of being too academic and explaining different concepts as we read them in a textbook, I'll try to contextualize it in our own gut-brain axis work and those of our colleagues in neurosciences. So first of all, let me explain a little bit of what we do at the Cambridge Parkinson's Research Clinic. We mainly have clinical cohort studies in which we recruited over 600 active participants. Most of them have Parkinson's disease. We try to recruit them within the first 2 years of diagnosis, so really early on into the disease process. And we see them at least every 2 years to have a sense of how the disease is progressing and presenting at several time points. Further to this, we also invite them then to do clinical trials, observational studies, and experimental medicine studies. But when they come to our research clinic, we spend about 2 hours with them, and we do quite a full comprehensive assessment. So we record clinical history, concomitant comorbidities that are other medical conditions other than Parkinson's or Alzheimer's, medication, and also we do motor assessments to check for motor impairment, cognitive function. So we do a battery of cognitive tests that I'm responsible for, and also some mood and quality of life questionnaires. All of the participants will also be asked to donate a blood sample from which we can extract serum, plasma, and DNA. We usually genotype for the main genes that are associated with Parkinson's disease, like Parkin's, CA, but also, for example, LRRK2, which is associated with both higher risk of Parkinson onset, but also inflammatory bowel diseases such as Crohn's disease. We also create awareness for brain donations, and that involves everyone with Parkinson's, Alzheimer's, healthy family members. We don't do this, of course, within our clinic. However, we create awareness for local brain banks that people might have in their geographical area, and we see people from all over the UK, and we invite them to consider donating their brain to science. I myself am a brain donor because I'm a romantic, and I'd like to think that even after I'm gone, I'll be still doing science somehow. So besides that, so everyone is invited to do those, but then according to specific studies, our participants might be invited to do extra procedures. We, depending on the study, we ask participants to undergo magnetic resonance scans, PET scans, or tau scans in order to see tau accumulation, which is a protein that is involved in Alzheimer's, and we'll talk a little bit more about that later. But we'll also collect PBMCs for further characterization of immune cells and function. We also, depending on the study, we don't do this too often, but we do have a couple of studies that we ask people to do a lumbar puncture and donate their CSF. CSF, they also call it spine tap. And the CSF is the liquid that protects around the meninges, insulating the brain, but also is a gateway into the central nervous system, in which we can see what kind of immune cells and nutrients and proteins are reaching the brain. We also ask participants to donate saliva, and we can quantify, for example, alpha-synuclein, which is again a protein involved in Parkinson's disease that I'll be talking later on, stool sample for microbiome sequencing, and brain tissue, in which we request to the brain bank, to the Cambridge Brain Bank, tissue from particular areas. In our case, we're particularly interested in Alzheimer's, so we're looking at the substantia nigra and doing techniques such as staining to look for particular cell types and cell function. But what is Parkinson's disease? Parkinson's disease is the second most common neurodegenerative disorder, only topped by Alzheimer's. And the key symptoms are slowness of movement, which we call bradykinesia, which makes it difficult for fine motor movements as well as walking. Tremor, although you don't necessarily have to have a tremor, and it can be as mild as this first image or as exacerbated as the second, especially as the disease progresses over time. And rigidity, which again impairs walking and daily activities. It is a neurodegenerative disease, so there is progression, and our own cohort studies show that at 10 years, about 70% of people with Parkinson's who didn't have balance problems or postural instability have developed them and are now having to use walking aids or wheelchair for mobility. About half of them as well, half of these participants have now developed Parkinson's dementia, which is a subtype of dementia with particular cognitive dysfunction. Each dementia subtype, so to speak, has a cognitive profile, and that's where originally my involvement came in the Cambridge Research Clinic. Underlying these symptoms and this progression is the loss of dopaminergic neurons in the basal ganglia, particularly in the substantia nigra. So as you can see here by this PET scan, a healthy control has a lot of dopaminergic neurons, as illustrated by the red, but as the disease progresses, there's a loss of these neurons, particularly an asymmetric loss, and usually there is the symptoms are more exacerbated in one side of the body over the other. And unfortunately, to date, we still do not have a cure, and symptom management is all we can offer to people with Parkinson's disease. And I guess this is where the gut comes in. So the gut-brain axis is implicated in Parkinson's disease in a variety of ways. So constipation is now well known and established as an official prodromal symptom. That is, ourselves here in this talk, if we are constipated as young adults, we are 3 times more likely to develop Parkinson's in the future. And this relationship is actually stronger than the association between inflammatory bowel diseases such as Crohn's or ulcerative colitis and Parkinson's onset, despite sharing some genetic overlap. And gastroparesis, or delayed stomach emptying, is also very common in Parkinson's disease, with cohort studies such as ours demonstrating a prevalence of 40 to 90%. And of course, this is not only an important factor for onset, so you're more likely to develop it, but also it affects how people experience Parkinson's disease. So because of gastroparesis, this is a gastroscopy, so an endoscopy in which a camera goes into the stomach about an hour and a half after someone with Parkinson's took their Parkinson's medication, which is levodopa. And as you can see here, an hour and a half later, this medication is still not absorbed, and thus delaying absorption by the small intestine. And so constipation is a risk factor for onset. It affects how I experience my Parkinson's and the relief that the current medication can give me. But my question in my PhD was, well, does it still affect the way people progress over time? And our work and the work of other colleagues with other cohorts has shown that, yes, so when we stratify in our studies, it was about 400 people with Parkinson's early in the disease process. And when we stratify them by constipation severity and adjust it for relevant confounders, including medication, cardiovascular disease, cognitive function at baseline, we saw that constipation was an independent predictor of how fast people would develop dementia. And our findings were validated by other cohorts, both in the States and in Italy, showing that constipation and the gut and gut health still affect how people progress over time. And of course, there's several mechanisms by which this might happen. We have microbial metabolites, we have the endocrine system, we have local and peripheral inflammation, which I will explain the mechanisms later on. But of course, today we're going to focus on the brain and the core processes within neurosciences and the techniques that we use to study to contextualize our own work in the gut-brain axis. So what is the brain? The brain is the body's primary control center. It's a beautiful mass of oh, Alex, should we stop or should I just leave the questions for later?
I would say we focus later. Just
Okay.
go through the talk, and then I'll chair, and then I'll point out the questions that were asked in the Q&A. So just keep going.
Yeah, sounds good. And so the brain is responsible for all really major functions, except interestingly the enteric nervous system. So if we sever, because of a gastric ulcer or other medical conditions, if we sever the vagus nerve, digestion is still independently achieved, which I find fascinating. But the brain is mainly, as I was saying, responsible for cognition. And so cognition can be divided into several cognitive domains. We usually associate it with memory, but of course there are several. We have executive function, which is mainly attributed to the frontal lobe, but also the basal ganglia, which is affected in Parkinson's. And so, for example, in Parkinson's dementia and even in cognitive decline in Parkinson's disease, we often see a decline in executive function. At the same time, or even before, we see a decline in memory. Memory is associated with the parietal lobe and the hippocampus, and is primarily affected in Alzheimer's, with memory and language deficits being the first cognitive domains in which we see an alteration, as well as visuospatial tasks, which are associated with the occipital lobe and which are also compromised in dementias such as Parkinson's disease. Of course, the amygdala is important, which is a subcortical structure, is associated with emotional regulation, pain and motor controls, which is in the motor cortex here in the frontal lobe, and involuntary functions such as breathing and heart rate, which are attributed to brainstem activity. And we know all of this because of the several techniques that we've been using throughout decades. We first started with lesion studies when we didn't have any imaging. It was people who had a stroke and the areas affected after having a stroke, or before work and labor safety regulations. We had people who had, like Phineas Case, which is a known example, that were impaled or somehow injured, and the areas associated with those injuries were then mapped or linked to the cognitive dysfunction and personality changes. Later on, we had electroencephalograms who measured the electrochemical activity in the brain, and then magnetic resonance imaging and PET. PET is a more metabolic imaging in which we inject participants with a radiotracer that binds to a particular protein or particular receptor in the brain as a proxy for a neuronal activity. And of course, animal models. And these animal models actually use all of the techniques that we also been using in humans. So for example, in Parkinson's disease or in Alzheimer's disease, we often still use lesion studies. So that's why, at least in Parkinson's, we call them Parkinson-like models, in which we inject a neurotoxin such as 6-OHDA or PTP or paraquat, which is a pesticide that is still used in certain states of the United States and is known to induce Parkinson-like symptoms. However, these models are not progressive over time. We also have genetic models in which there is overexpression of this protein, alpha-synuclein, that we'll be talking later on. And we also do MRIs and PETs to animal models as well. But what is happening inside the brain? So the brain is mainly composed of neuronal and glial cells. Neurons being, of course, the star of the show. You know, they form the basis of the nervous central nervous system. We have 86 billion of them, and they're mainly divided into the soma, the body in which the nucleus is, as well as other important organelles, the axon, in which the information is relayed, and the dendrites, which are these tree-like structures that receive input from other cells. Now, these neurons, although being the stars of the shows of the CNS, actually are highly dependent on glial cells like astrocytes, microglia, and oligodendrites, which are in the same number, about 86 million as well, which I think just illustrates the importance of them, without which astrocytes, neurons couldn't function. I'm going to start first with astrocytes. So for example, astrocytes are these supporting cells that liaison between the blood vessels in the brain and the neurons, trying to match the metabolic demands. So for example, if neuronal activity is increased, this metabolic demand is conveyed to the astrocytes, who sense the neural activity and request from the blood vessels the inflow of nutrients, particularly glucose, ATP, and ATP as well as, and this is particularly important, although this is an organ which is, 2% of our body weight, is about 1.5 kilos. It actually consumes about 20% of our glucose, recommended daily intake, which is about 120 milligrams per day. Astrocytes also coordinate with immune cells, which is the microglia that I'll talk in a minute. So when there is damage, injury, or some kind of distress sensed in the neurons or identified in the neurons, then astrocytes, along with microglia, will signal the vascular system to recruit or traffic into the brain peripheral immune cells such as lymphocytes and monocytes. And they also help clear out neurotransmitter, and we'll talk about synapses in the next couple of slides. I forgot to mention, but of course this is very important because this is another technique used in neurosciences that is associated with the neurovascular coupling. So a lot of the cognitive studies that we used in neuroscience rely on this beautiful relationship between neurons and astrocytes. So as in a particular task, the neuronal demand is increased, and there is an increase of blood flow exactly to recruit the glucose and the oxygen levels and the ATP levels. There is an increase of blood oxygenated signal that we can detect by functional MRI, and this is how we know which areas of the brain light up within a certain task. There are many limitations to this technique, but it is still one that is used today, and we don't use it in our lab. We use it more in PET scans in our group. Another important concept that I'd like to discuss within still the astrocytes is the blood-brain barrier. So the blood-brain barrier is a membrane that is responsible for homeostasis and the selective trafficking of these nutrients, but also the immune cells that can in and out, and also hormones and medication. And a lot of the drug targeting work is revolved around making sure that the molecules have the right size or the appropriate chemical properties to go through the BBB. Having said that, in neurodegeneration, like Parkinson's, like dementia, the tight junctions of the blood-brain barrier are compromised, and so just like we have a leaky gut, we can also have a leaky brain that happens in normal aging, but is particularly exacerbated in Parkinson's disease because they're particular, and Alzheimer's because there are particular tangles that compromise this barrier. And so when this barrier is compromised, and when there are toxins and cells that shouldn't be in the brain are in the brain, then we rely on this immune cell of the brain. You've often heard about them as the resident cells of the brain, the microglia, which survey the environment for distress signals. They signal pro-inflammatory or anti-inflammatory cytokines to recruit the astrocytes, recruit the peripheral immune system. They are macrophages, so they phagocytose debris and also synapses. So if synapses are connections between neurons and other neurons or other cells, if you have redundancy or if the synapse is not functioning properly, the best efficient way to increase homeostasis is just to prune them. And of course, we also have the oligodendrocytes, which are cells that are responsible for producing myelin, which is a fatty insulator that goes all around the axon, and basically it's protecting the signal, the electrical and chemical signals, from getting lost. So, of course, this is an oversimplification of what happens in the brain, because actually it's much more complicated. So, a neuron can be connected with more than 10,000 other neurons. And actually, the Allen Institute did an amazing feat, and they were able, through high-resolution imaging and computational analysis, to recreate just 1 cubic millimeter of the primary visual cortex of a mouse. It's very interactive, and I encourage you to to visit this website. And of course, within this milliliter, why do I say milliliter? Millimeter, it includes over 200,000 cells and 523 million connections. In our lab, we don't do this high computational imaging thing, research, but we do immunohistochemistry and staining. And so for us, the microglia that are perfectly represented here actually look like this, as well as the astrocytes. And now I claim really no knowledge of this technique. However, for example, I was an author in this paper because I was one of the team members responsible for recruiting and doing data collection and motor and cognitive assessments to some of these controls and some of these very kind and brave people with Parkinson's who donated their brain to science. And although I am not a biologist, I've done my fair share of ELISAs and wet lab work. I'm not a biologist. I did do a little bit of contributing, so to speak, to these sort of projects, and I just wanted to share it with you. So this is another more innovative technique within neuroscience. The Songlag at Princeton University developed a game in which it's a citizen or crowd science in which you can learn how to map different parts of a cell. And so the way it works, because I did this over 10 years ago for a couple of months, and they teach you how to identify particular structures with colors. If you successfully identify a few, then you are given, I don't know how many dozens to identify, and then several iterations of them. And then when there's a congruent agreement between gamers or citizen scientists, then it is established as, a neuron or an axon. And with this, they're hoping to again map retinal neurons, I think, in mice. But I was talking a lot about neurons and how they communicate and how they convey information and how we can think and process and make decisions. But what is happening? How is this communication happening? Well, we rely essentially for these processes on the synapse. Synapses are junctions in which a neuron can transmit information, usually either via electrical information in ion flow or chemical. Chemical are the most common. And the sending neuron, we call it the presynaptic neuron. And of course, neurons can communicate not only with other neurons. Again, this is an oversimplification because we always think neuron to neuron, but neurons communicate with mainly any excitable tissue. So we're talking about neurons, we're talking about glands, muscle, and of course other cell types like enteric cells. So the way this works, in summary, of course, is when an actual potential is achieved, there prompts the release of vesicles that are filled with neurotransmitters, which we'll talk about in a minute. And these neurotransmitters are moved into the synaptic cleft, which either by diffusion, meaning they just float around and then they're taken up by microglia and astrocytes, if they're not binded to specialized receptors. And these receptors might be specific to a particular type or types of neurotransmitters. And of course, they can also rely on transporter proteins to move, although this is a very small junction. And what do they transport? Well, they transport most neurotransmitters. The ones that are most commonly known and I'd like to emphasize is of course dopamine. Dopamine is associated with focus and cognition, decision making, but also reward. This regulation of the dopaminergic system can include, for example, if it's overstimulated, obsessive compulsive disorders or impulse control disorders, in which you can have gambling or obesity, and an under-regulation of this dopaminergic system through, for example, neuronal dopaminergic loss is associated with Parkinson's disease. The serotonin is also a very well-known neurotransmitter because it's associated with mood, and it's, I think, called a happy hormone. Of course, it's associated with a plethora of other cognitive processes, and its depletion, although this is a bit controversial, but its depletion is associated with disorders, mood disorders like Alzheimer's. And of course, you also have acetylcholine that is associated again with cognition, but also voluntary movement and involuntary movement. And for example, in Alzheimer's, there's a depletion of the cholinergic pathways. And although all of these neurotransmitters are associated with the brain, and that is mainly the context in which we know them, actually, I think about 90% of serotonin is produced in the gut, and about 50% of dopamine and acetylcholine is also produced in the gut, although these molecules have a very short half-life, so we assume they are just acting on a very peripheral and local function. However, they're incredibly important for the normal functioning of the brain. Let me see why I can't move the slide. So again, how do we have this basic understanding of how cognition works at a cellular level? Well, we have several in vivo and in vitro techniques. Some of the in vitro techniques are listed here. And for example, again, I find it beautiful that apparently this is a vesicle for full of neurotransmitters, and we rely on microscopy and immunohistochemistry, which is staining different cell types, in order to have a comprehensive view of how this processes work. Again, in the group, we use mainly in vivo techniques, and whilst we can't image dopamine or serotonin per se, we can image the transporters that I mentioned that are responsible for taking dopamine, for example, either back into the cell or to help with the transport into the receptor binding. And so, for example, when we as researchers want to try have a sense about the dopaminergic loss in people with Alzheimer's, or there's question about the diagnosis of Parkinson's disease, because as you can see, slowness of movement and rigidity and even tremor can have an overlap with other movement disorders or even normal aging. So in cases where that is put into question, people are often invited to do, for both in the NHS and for research purposes, a PET scan, which binds to DAT, which is the dopamine transporter protein. And as you can see here, in an abnormal DAT scan, usually associated with people with Parkinson's disease, you have an asymmetric loss of this DAT, which, because it is in the presynaptic neuron, if it's lost, if there's less density, then we assume there's neuronal loss of dopaminergic neurons. I've just mentioned briefly this overlap between what is considered normal aging and neurodegeneration, and so how sometimes it can be hard to distinguish between the two. That is because it's a spectrum. So neurodegeneration, by definition, includes a progressive loss, an irreversible loss of neuronal either structure or function that often leads to cognitive and motor dysfunction. And why this particular point is important is because we'll see later on, because sometimes you do have loss of neuronal structure or neuronal dysfunction, and yet people present no symptoms. And we see this not only with PET imaging, but also post-mortem imaging. They're often progressive and irreversible. However, they can assume a step-like stage where you can have small regressions in terms of symptoms or dysfunction, but on the long term, they are sadly irreversible. And again, as I was saying, they are associated with aging. They're pathological aging. However, they overlap in many of the symptoms. And we now know a few predictive prodromal markers. We've mentioned constipation, but for example, in Parkinson's disease, it is often seen also accompanied with REM sleep behavior disorder. This is a sleep disorder in which people talk, scream, shout. They have very active dreams, and they are not mobilized in bed, and so they'll often punch and get out of bed. And sadly, according to cohort studies, and we do also have an RBD clinic in our group, about 90% of people with this sleep disorder will develop Parkinson's. So much so is that if you go to your GP and then you do a PSG, a polysomnogram, in which you are identified as having REM sleep behavior disorder, you are informed that you are at an incredibly high risk of developing some form of Parkinsonism. But of course, all of these can exist without the progression to a neurodegenerative disorder. And so the important part is, when does it become pathological, and where is the best time within this spectrum to intervene? Well, I guess here is a good point to say then, what is happening in the brain then? Depending on what are the mechanisms that are driving the different neurodegenerative diseases. So for example, in Alzheimer's, at the organ level is the hippocampus, which is the brain structure that I mentioned right in the beginning, that is associated with memory function. And at the tissue level, there are abnormalities in the hippocampus because of the loss of pyramidal neurons, again in the hippocampus, but also in the cortex and the amygdala. And this loss is attributed to the dysfunction of both amyloid beta and tau proteins. So tau proteins are microtubule proteins that are associated with structural function in axons, and of course they can form fibrillary tangles that turn toxic when they do. Same with amyloid beta. It is important for hormonal storage and neuronal support, but then again it can aggregate into toxic forms called amyloid plaques. In Parkinson's disease, it is known to affect the basal ganglia, particularly the substantia nigra and alpha-synuclein, which is a protein that is involved in vesicle and neurotransmitter transport. Again, also hyperphosphorylates and aggregates together into Lewy bodies, which are toxic to particularly dopaminergic neurons. In ALS, we have the motor cortex, a terrible disease that affects the motor neurons that are responsible for both voluntary and involuntary motor movements. And like Huntington's disease, they are mainly genetic. For example, Huntington's disease is a dominant disease. So myself, if I had a Huntington's, I would have a 50% chance of having children with the same disease. And it is basically associated with mutant huntingtin that accumulates in spinary medium neurons. Again, this is an oversimplification, because actually they're all of these mechanisms that are associated with NDDs, or neurodegenerative disease, such as inflammation and neuronal death, are present in all of them. So we see through PET imaging, for example, we see, and post-mortem studies, we see amyloids plaques in the brains of people with Parkinson's disease. We see inflammation in Alzheimer's, we see inflammation in Parkinson's, we see an increase in RRS in all of these diseases. And so the picture is a little bit more complex. And I just made this table a couple of days ago to show you exactly one of the challenges that we face when we're studying neurodegenerative disease. So, for example, it's very common to see brain atrophy, and that is expressed in, again, we say neuronal loss, but of course astrocytes and other glial cells are also affected. But over 40, we do expect a 0.4 decline, and apologies to those over 40 in this meeting for the horrible and deterministic information, but over 0.4 people per year will experience some form of brain loss. This is, of course, just exacerbated in the different neurodegenerative diseases. Same with vascular function. That is why exercise and hypertension and hypercholesterolemia are important, because it is very common to see small vessel disease and white matter hyperintensities in people over 40. But of course, this again is extremely pronounced or accelerated in NDDs. Same with neuroinflammation. It is very, and we do this, we do a lot of characterization of healthy controls over 50 years old, and we see microglia senescence, we see a loss or a decrease in T lymphocytes. There's a whole body of literature on inflammaging, which perhaps this was touched upon in the first GIBA session. And of course, we see this again in much higher levels in different neurodegenerative diseases. And the same with the exact proteinopathies that I was explaining in my previous slide. We also see it in Parkinson's disease, of course, just not at the same level that we see in pathology. So what are the caveats and challenges that we're currently facing? Several of them. So if this is a spectrum, then when is the disease achieved? And I'm not asking just on behalf of us neuroscientists who are within the clinical neuroscience field, and we need to know who to recruit. It trickles down to stratification and the cell lines that what biologists and wet lab scientists use. It's important also to note that, for example, sometimes, and I work with neurologists who don't exactly, they can't really confirm whether someone has Parkinson's or Alzheimer's, because it might be just a mild cognitive impairment, which will progress to dementia or stay stable over time. And sometimes the best thing that we can do for a moment is monitor, which sounds awful, but in a lot of times, both in research and clinic, we'll just wait a year and see whether they progress. And it's the progression that sometimes is the key towards a proper diagnosis. And nevertheless, for example, in Parkinson's, about 5% of cases diagnosed with Parkinson's are diagnosed within the next 2 to 3 years with a different movement disorder. So where we draw a line in diagnostic accuracy is very important. Another caveat that we have trying to understand this disease is animal models. As I was saying in the beginning, animals don't really get Parkinson's. Animals can get dementia, cats can get dementia. They can have cognitive issues, but they don't have Parkinson's. And so what we do is we lesion them in the substantia nigra, which can give animals Parkinson-like symptoms. We can create knockout or knock-in models that overexpress alpha-synuclein and create a proteinopathy. But again, this progressive loss is not really well captured. And so whilst it's extremely important to understand the mechanisms and the cellular mechanisms, it's not a true replication of disease. And we also have the toxicity model. So this is a PET scan. And again, the higher the red color, the more dopaminergic neurons we have, and these are called SWEDs. So these are scans without evidence of dopaminergic deficits. So these are people with Parkinson's disease for which we do a scan, and actually they don't have dopaminergic loss, and we still can't exactly explain what is happening here. And we have the opposite as well. We have at post-mortem and in PET imaging healthy controls who lived a full life and do very well in their cognitive testing, and actually they're quite their brains is what do you call it, presenting with a lot of amyloid plaques. And what is the resilience process? We had a postdoc in this group that now has moved to Oxford, but I found his work fascinating, and he'll work exactly on neuronal vulnerability. Why is in Parkinson's disease dopaminergic neurons affected, but all other neurons are of course affected, but not to the same extent where you lost more than 80%. Rule of thumb is that if 80% of your dopaminergic neurons are lost, then you're presenting with symptoms. And even within Parkinson's disease, even within Alzheimer's disease, there's so much heterogeneity in presentation. So this is one of the cohort studies that we did, and it's the same result. So at 10 years, about 70% of people had postural instability. 50% were had Alzheimer's, had the dementia. Sorry, but about 25% actually were doing quite well at 10 years. What is the molecular? What is the biological protective factors that were keeping them from this accelerated progressions? And some people have tremor and some people don't. Some people have behavioral issues in Alzheimer's and some people don't. Could there be subtypes? Could be one of the reasons why it's keeping us from advancing to new and effective drug therapies is because we're clumping together several diseases into one. Well, we think that the gut might be explaining, within the group, might be explaining this heterogeneity in presentation and progression that we see. And of course, there's several mechanisms by which this might happen. We have the vagus nerve. So we know that alpha-synuclein is, let me just see the time. I don't want to, okay, I think we're good. Alpha-synuclein is also present in the gut. This is work done with biopsies or colonoscopies. And for example, we know that after a viral infection, this was a study done with children, that after a norovirus, there was aggregation of alpha-synuclein in their colon. Alpha-synuclein aggregation is associated with both constipation, but also local inflammation. And just like we have permeability in the blood-brain barrier, we also have permeability in the gut, and the trillions of microbes who do incredible work to keep the homeostasis might translocate in the periphery. And our own studies show evidence that although we think the blood should be a sterile medium, actually we saw both LPS and PGN in the periphery, and we'll talk a little bit more about that later on. So the immune-brain axis might be implicated, because then if there is a slow translocation of microbes who are benign and are endogenous, what might happen is this might be explaining why we see a low chronic inflammation in people with Parkinson's disease. And this low chronic inflammation perhaps is being trafficked into the brain, which further activates microglia, and then is leading to dopaminergic loss because they're macrophages and perhaps they're being overstimulated. This is the working hypothesis: is constipation, is it associated with both peripheral inflammation and central nervous system inflammation? Well, most of this work has been done in mice, but recently we just published that study. This is a ligand that is injected in people with Parkinson's disease and binds to TSPO, which is a transporter of the dopamine in that synaptic cleft that we talked about earlier on. These were about 30 people with Parkinson's very early on into their disease process. About 40% were constipated, and I was expecting some correlations between self-reported constipation scores and neuroinflammation. I was not expecting that more than 20 ROIs, and including whole brain analysis, showed a positive correlation between constipation and neuroinflammation. Oh, sorry, did I say dopamine? I meant microglia. Apologies. TSPO is a marker of microglia. And then we looked at the CSF of these same participants, and we saw that the higher the constipation scores, the higher the lymphocyte count in the CSF, and pro-inflammatory lymphocytes in the periphery as well. We would now, because this was the first and that I know the only study showing this relationship in humans, we are currently now recruiting and validating this same study with another ligand from microglia, DPA714, in more objective measures of constipation, and that's to obtain a whole gut transit time. So because there's a lot of underestimation of constipation, and so with a whole gut transit time, for those of you who work with animal models, it's the same thing that we do it with little mice. So we ask someone to, after going to the toilet to empty their bowels, we ask them to consume a blue dye, icing baking dye, and write down the date and the time in which they consumed it, and the date and the time in which they poop blue in this case. And actually, although some of our participants, we published this results, I think now 2 years ago, we did it in over 80 people with Parkinson's and healthy controls. And although some people with Parkinson's and some controls, although much less, were telling me that they were not constipated, they were pooing every day. Actually, they were pooping blue 7 days later, 8 days later, up to 9 days later. I mean, they had constipation in terms of delayed transit time, although they were going to the toilet every day. We are also doing methane breath tests because constipation is associated with archaeal overgrowth. Archaea are methanogens, or many of them are, and so methane is used in gastroenterology as an objective measure of constipation, and this work is ongoing. I think I still have 6 minutes. I'm sorry if there's not a lot of time for questions, but the take-home message is gut dysfunction, particularly constipation, is not only a risk factor for the onset of neurodegenerative diseases, but also a risk factor for progression, meaning perhaps it's a targetable, it's a target that we could exploit in order to prevent or a disease onset or delay disease progression. Aging, both normal and pathological, is a complex multifactorial spectrum, which again, the gut seems to be heavily involved in. And neurodegenerative diseases allow us to understand this bidirectional relationship. A lot was not said, but for example, alpha-synuclein propagation has been found to travel up and down the gut. And for example, people who have gastric ulcers and have to sever the vagus nerve are less likely, at least in the short, longer term, to develop Parkinson's. So a lot is being done, and I thank you for your time and attention.
Excellent. Thank you, Marta. So we're gonna take some time for questions. We'll probably do it 10 minutes or so. David, I guess your talk will be about 40 minutes, right? Yeah, okay.
Yes.
So we can start about 11:05 or around then. So we already have some questions. I asked people to start thumbsing up specific ones they might be interested. So the first one that has the top number is from Mary Ellie Louise. So thanks for the great presentation, really interesting. I was wondering what gut metabolites, if any, are known to be associated with Parkinson's or neurodegenerative diseases.
Oh, that's a very interesting question. So, for example, it's not a metabolite per se, but I am interested in the role of ghrelin. So ghrelin is produced in the stomach and it's associated with gut motility and satiety. However, it can cross the blood-brain barrier, and we see that it suppresses microglial activation and protects dopaminergic neurons when we stimulate them in vitro with LPS, which is a bacterial fragment. And so we're looking into that, and we are quantifying ghrelin levels in people with Parkinson's, people at risk of developing Parkinson's, so that's the REM sleep behavior disorder, and healthy controls. And what we saw during my PhD, and again, we're doing this in a little bit more in-depth manner, is that people with Parkinson's had really low ghrelin levels compared to controls, and those with RBD, so those at risk of developing Parkinson's, have even lower. And this is across multiple time points, meaning it might be a phenotypic trait rather than a state. And so we're going to look at that a little bit more. We also have, for example, the work of Samson et al. I think they published in 2016, where it's not necessarily related to Parkinson's, but of course, short-chain fatty acids are can be protective of dopaminergic loss. But I don't think, in the sense of a detrimental effect, I'm not sure of any particular metabolite that is associated with Parkinson's. Is more the opposite.
Okay. Another top question. Thank you. Can you explain a bit about the mechanism underlying the positive association between constipation and Parkinson's disease? Are there any evidence that diet, such as fiber intake, can improve PD symptoms through alleviating constipation?
So we have a sense of some of the mechanisms, but that might be explaining this underlying relationship between constipation and Parkinson's. It could be that constipation is associated with gut permeability. So the more constipated you are, the more mucosal integrity dysfunction you have, and more local inflammation you have, the more alpha-synuclein phosphorylates in the gut. And we also know that fiber intake can be protective. So, for example, there's plenty of studies with the Mediterranean diet showing that it is protective in delaying. So you have the same prevalence of Parkinson's disease in Mediterranean countries compared to, for example, England, but the average onset, so the average age at diagnosis is slightly later. So whilst they are related, the relationship, of course, is not linear and direct. So it seems to help delay progression. But what is it within the Mediterranean diet? And of course, you also have to think that fiber is not a fit for all. So a lot of people have are in the spectrum of celiac disease and are not aware of them, nor aware of it. And so a high fiber diet might not be necessarily good for them. The same thing with, for example, flax seeds. If you have diverticulitis, you can actually inflame and induce stomach cramps and bloating. So I think the field of both neuroscience and also gastroenterology is moving towards a more personalized precision medicine studies. And that's exactly, for example, what we're doing with my ghrelin studies. So we're giving something as an experimental medicine study to people with Parkinson's and controls. And if I can increase ghrelin levels in controls and not Parkinson's, we're thinking, what does that mean? Does it mean that this doesn't work, or is there some sort of ghrelin resistance mechanism that we see in insulin for diabetics, but in Parkinson's? And of course, ghrelin is again associated with gut motility and gastroparesis. So the endocrine system seems to be heavily involved in this as well, and that would be separate from diet altogether. So there's a lot of mechanisms, but do reach out via email, and I can send you some really nice papers on this.
Okay, so another question. Have you considered looking at the bacterial extracellular membrane vesicles of people with constipation and neuroinflammation? Since those vesicles are known to cross the blood-brain barrier, it might be another tool for understanding this link. I'm not sure, Mart, if you have done or looked into this.
No, sadly, I have thought about it, how cool it would be to do. And if you want to establish a collaboration, I'm more than open to do this by providing samples. But sadly, it's very much beyond my expertise. But I think we're definitely you're onto something there.
Yeah. Maybe you have touched upon this. There was another question about constipation and what severity level is constipation associated with Parkinson's disease, and can you share how this was measured?
Yes, that's an excellent question. So that first paper that I showed you, it was a self-report and a single item questionnaire of how constipated are you. So that would be no constipation. I'm constipated and I employ methods to relieve my constipation, and those are beneficial. So if I increase my fiber intake, if I take laxatives either twice a week or every day, I'm comfortable. And then the last stage of constipation was despite employing methods, including enemas, I'm very uncomfortable with, and those are not beneficial, and I'm very uncomfortable with my constipation. So I stratified people with Parkinson's within those three categories. Now we're replicating this, but using whole gut transit time and using methane levels, as well as self-reported measures. So hopefully that will give us a better insight on their constipation in a more objective way, and not underestimate or overestimate their scores, their constipation scores.
Okay. Is there an understanding of why animals cannot get Parkinson's? Is it just structural differences, or is it something worth looking into to understand the onset in humans?
I don't know either. I know there's a lot of research on neuronal vulnerability, but I'm not quite sure why. Maybe David knows.
No.
No.
No. Okay. You mentioned that the mouse models of Parkinson's are not progressive. Does this suggest that you do not see the expected prodromal symptoms in mice? For instance, in genetic models of PT, do the mice develop constipation?
So the mice do develop constipation, and for example, if you inject alpha-synuclein into the brain of a mouse, you will see alpha-synuclein deposits just a few days later in the gut. And actually, that is how we know there's bi-directional transport of toxic alpha-synuclein propagation via the vagus nerve, and that it does induce constipation. And we measure, well, I say it's a very royal we because I don't do this, but researchers who investigate constipation in mouse models do very similar techniques to ours. They weigh the mouse stools. They see how often they defecate, and they also use blue or red dye to measure transit time.
Okay, and we'll take one last question. So this is the one with the one thumbs up. Thank you for the resourceful presentation. Really interesting. I have a question about inflammation status. Can you see a difference between Parkinson's patients who have constipation and those who don't in terms of inflammation status?
So I've now stopped sharing my slide, but we do. So we have a well-defined low chronic inflammation in Parkinson's disease that seems particularly heightened during diagnosis and seems to trickle down. So perhaps it's reactive, we don't know, but we do see a decrease of lymphocyte count in Parkinson's disease. And in the studies that we published, we saw higher lymphocyte count in the CSF. So that's already within the central nervous system. There's some theories that say that perhaps that it goes from the periphery into the CSF. And there's other people in my group who do flow cytometry and are trying to further characterize this immune dysfunction and upregulation in Parkinson's disease. But it's well characterized, and I invite you to read the many studies that are done in this field.
Okay, excellent. Thank you very much, Marta, for that excellent overview and presentation. We're going to now move on to the second talk from David Sanderson. So David is a professor in the Department of Psychology at the Durham University. His research focused specifically on learning, memory, and behavioral neuroscience, including how animals encode and retrieve information. His research uses experimental models to investigate both the neural and cognitive mechanisms underlying memory and related learning processes. So David is going to give us an overview of how he does behavioral and cognitive studies in mice. Over to you, David.
Thank you. I take it you can all see my slides?
Yes, all good. Perfect. Thank you very much.
Yeah, thanks. So I'm a hello everybody. I'm a behavioural neuroscientist, and I'm in the Department of Psychology. So in some sense, I think of myself as an experimental psychologist. But I work with rodents. So what I'm going to talk today about today is how you go about measuring cognitive function in animals and relate it to particular brain processes. So in terms of behavior in an animal, if it has inflammation or some kind of challenge, there might be all sorts of behavioral consequences. But if you want to link it specifically to cognition and learning and memory processes, then you have to find particular ways of going about measuring those learning and memory processes. And what we know from the study of learning in animals from the last 100 years or so is that actually there's very specific psychological processes that can be dissociated when you look at performance of learning and memory in animals. And unsurprisingly, those very specific processes are also can be dissociated at the neural level. So different brain areas seem to be involved in different processes, and within those brain areas, various neurotransmitters or receptor subtypes are involved in determining what the animal learns and remembers in particular situations. So I'm going to show you a bunch of methods for studying learning and memory and how they relate to particular brain functions. The talk may take some kind of rapid twists and turns, but they all each example illustrates how we'd go about trying to be able to isolate a particular psychological consequence of some kind of neural manipulation. So to start off with, as Marta mentioned in her talk, when we think about learning and memory and the role in the brain, we think of the hippocampus, this structure here. So the hippocampus, certainly in humans, we know it plays an important part in remembering information. And probably the most important study in informing us about the role of the hippocampus was the study of the patient Henry Molaison, referred to as HM during his life, who he suffered from epilepsy, and he had surgery to alleviate him of the epilepsy, but an unwanted consequence of the surgery was that he had significant damage to the hippocampus. And what was so fascinating about Henry Molaison was that he was absolutely fine in many respects, and he could go about his everyday life, but he just had no new memory of events. So he suffered from, he wasn't able to acquire, seemingly wasn't able to acquire new memories. So he would meet various people day to day and then have no memory of those particular meetings. However, what we know is that he was able to learn new things under certain circumstances. So it's well documented that he was able to acquire new motor skills, and he could learn about particular information. But when actually asked about anything, as in asked whether he could do a particular motor function, he would say no, because he had no memory of actually doing these things. So what we've learned from the study of HM is that the hippocampus in humans is important for episodic memory, remembering particular episodes of experience, the kind of what we might refer to as the what, where, and when of a particular episode. In animals, we can study various aspects of episodic memory using particular methods. So we know in rodents that when you damage the hippocampus, like Henry Molaison, they're able to do all sorts of things, and they are able to learn lots of things. So in terms of kind of standard Pavlovian conditioning procedures, maybe with lights and auditory cues, They can learn these associations fine, but they fail to learn about spatial environments. And there's various suggestions for why this may be, about what the role of the hippocampus is in spatial learning. One idea is that you need the hippocampus for representing the spatial environments. And you may know about the study of place cells. So John O'Keefe at UCL has been the person who's pioneered this, but you find cells in the hippocampus that fire specifically to certain spatial locations in an animal's environment. Well, in order to study spatial learning in rodents, a very influential procedure that people have used is the Morris water maze. So Richard Morris in Edinburgh developed this procedure, and it's very good because it provides a very controlled test of spatial learning. And specifically, what it requires animals to do is to learn about the allocentric cues, so the cues outside of the immediate maze that the animal is put into, and it has to learn about those cues and the spatial relations between them. So what it really is a giant tub of water, and a mouse or a rat, in this case a picture of a mouse here, is required to swim round the maze and to locate a hidden platform. So here you may be able to just see that there's a white circle there. There is a platform which is just underneath the surface of the water, about a centimetre or two. And rats and mice, when they're placed into the maze, they don't know where the platform is, and they swim round randomly. Quite often they will go around the outskirts of the maze to begin with, and then they'll go further in, and then they'll happen upon the platform. So rodents are natural swimmers. They will readily do it, but they will like to sit on the platform. So the platform is rewarding. And so here you can't see in this picture, but there are cues around the room, posters, and there may be cupboards and doors and various other visual cues around the room. And the animal can learn where the location of the platform is within this pool by using those cues. And there's various control methods that you can use to demonstrate that is exactly what animals are learning. And that's important because the hippocampus seems to be important in representing the environment. So if you give an animal an alternative strategy, such as learning about egocentric body turns, then you may not need the hippocampus for those procedures. So here are the results from a paper from some years ago with David Bannerman and colleagues, where mice with hippocampal lesions were trained on the Morris water maze task. And what we're measuring across blocks of training trials here, is the path length that they took to locate the hidden platform. And as you can see, that control animals that had a sham lesion were able to find the platform and take shorter path lengths over training, whereas these path lengths remained high in mice with hippocampal lesions. Here, just to illustrate, here's an animal that has an intact hippocampus. These are coronal sections going back through the brain from the front to the back, and you see the hippocampus coming through here. And here's an animal that has a lesion, so they've had a neurotoxic lesion, and you can see that the hippocampus is largely missing. Now, the problem with this is that the fact that sham lesion animals are doing better than hippocampal lesioned animals could be that they're learning about the spatial location, but it could be because that they are just adopting an efficient search strategy. So you may think that you take less time to locate the platform simply because you become efficient at searching, rather than because you know where the platform is. Well, what we do to be able to be sure that animals have learned the spatial location is that we can have a probe trial where we remove the platform. So here on the probe trial, the platform would be missing, and we just see where the animal searches over a period of time, say, for example, 1 minute. And if the animal knows where the platform is, it should search predominantly in the area where the platform used to be. So what we do typically is divide the the maze up into quadrants and see whether it shows a preference for searching in the quadrant where the platform used to be. If it just has some search strategy, say for example, the animal may learn that the platform doesn't know where the platform is, but it knows that it's a certain distance from the edge of the pool. Well, then it will swim in a circle like that. So it should show equal amounts of time in the different quadrants. And of course, what animals do very sensibly is that control animals show a preference for exploring the goal quadrant, the quadrant that used to have the platform of the hidden platform, whereas hippocampal lesion animals don't. So they do seem to show a deficit in being able to find the location of the platform. And of course, as I said, that's in contrast to various control procedures where you show that hippocampal lesion animals can learn about other non-spatial things, but they can't seem to learn about this spatial task. A much simpler variant of this spatial learning task is something that you could do would be in a Y-shaped maze. So here we have a Y-shaped maze, and there'll be food in this particular goal end of this arm. And we might start a mouse or a rat from either of these two start arms, and it has to learn to turn to find the reward, the food reward at this particular end of the maze. Now, obviously, it can't learn just by body turns, because if it starts here in this particular start arm, it has to turn right, whereas if it starts here, it has to turn left. So it has to learn about the particular spatial location that is rewarded. And you do various things, such as you can rotate the maze whilst keeping the room cues constant to be able to rule out that the animal's using some intramaze cues. And what this figure shows here very strikingly is that overtraining, control animals become very proficient at over 90% correct, but hippocampal lesion animals never go above chance. They fail to learn this task. And of course, this is all consistent with the idea that the hippocampus is its role in spatial memory is probably representing spatial locations. However, another take on this is that actually that during spatial navigation, animals have to remember where they've been in order to search efficiently. So if you're navigating a particular location, it's very handy to be able to have a record of where you've been recently in order to be able to get to your particular goal location. And this is illustrated by this procedure here in the radial arm maze. So here, imagine that we've got we're looking down upon the maze, and an animal would be placed in a central platform, but it can run down these arms radiating out from the middle of the maze. Now, on the start of a trial, it could be that there's food at the end of every single arm. So if an animal is going to search efficiently to collect all the food that's available, it should run around, get food, and not return to arms that it's previously entered. So to do this in as fewer arm choices as possible, you shouldn't return to where you've previously been. So we refer to this, or people have referred to this as spatial working memory. Working memory in the sense that you're having to keep information online. You're having to have some kind of short-term store of what you've done that's relevant to your particular current task. So to give you an example of what might happen is that an animal may be placed in the middle, and they may choose this arm to go down first, and they can eat the food at the end of this particular arm. So subsequently, when they return to the middle, this arm doesn't have food, but the other arms do have food. So maybe on its next choice, it goes to this arm and it chooses to eat food there. But then on its third choice, if it then returns back to this arm it's previously been in and there's no food reward, we would say it's made a working memory error. Now, unsurprisingly, hippocampal lesioned animals are impaired in this task, but we don't know whether it's because, or we certainly didn't know the people who did this originally in the 1970s didn't know whether it was because the hippocampus is important for representing the spatial locations that need to be remembered, or whether it's put specifically because of the working memory properties of this task. But I'll come on to show you some data that shows that we can dissociate these particular accounts at particular levels. So here's another variant on the working memory procedure, which is in a T maze. We've had a Y maze, now we're having a T maze, so it's shaped like a T. And all that happens in this procedure, the T maze rewarded alternation procedure, is that an animal is placed in this start arm, and on the first run of a trial, when it runs down the arm, one of these goal arms is blocked, so it can't go down here, so it has to turn down to the left goal arm, and it has some food reward. So we've forced the animal to go down there and turn left. However, on its second run, the choice run, when it runs down the start arm, it can now turn left or right. But importantly, it will only be rewarded for turning right and not be rewarded for turning left. So the animal needs to remember where it went on its sample run in order to get food reward on its choice run. So just like in the radial arm maze, it has to remember where it's been recently. So here are the data for performance on this in various different groups. So in this first section of this graph, here are control animals who perform very well at this task, way above chance at 80%, and rats that have a complete hippocampal lesion, they show chance performance. But what we know is that this seems to be entirely due to damage to the dorsal hippocampus. So if you have a lesion that's limited to the dorsal hippocampus, you also show chance performance. Whereas if you have a lesion that's just for the ventral part of the hippocampus, it seems to be without effect. So this seems to be a specific role for the dorsal hippocampus. We see similar effects in mice. So mice that have lesions of the hippocampus, just like rats, show a similar deficit. But also, here are some data for some genetically modified mice that lack the GLUA1 subunit of the AMPA receptor for glutamate. So these mice are interesting because they show impaired hippocampal synaptic plasticity. So they have an impairment in long-term potentiation of synaptic plasticity within the hippocampus, potentially a mechanism for hippocampal dependent learning. And they show an impairment on this spatial working memory task. And what's very interesting is that when they're trained on tasks like the water maze task, which you can think of as a reference memory task in as much as the animal just has to acquire association between a location and an outcome, they are fine on those tasks. So they don't show the hippocampal deficit that hippocampal lesion animals normally show. Now, performance in this task may suggest that you have to learn that you have to alternate your turn in the choice run. But actually, what we know is that animals don't need to learn this. They show this behavior spontaneously. So this is some kind of innate memory that animals have for particular locations. They don't need to acquire an alternation rule or some working memory rule. They just show this behavior where they have a tendency to not return to previously recently experienced spatial locations. So in this task, we've got the Y maze again, but here animals aren't rewarded. They're just allowed to explore two arms of the maze in an exposure phase of this experiment. So the animal's placed in the start arm, and it can run between these two arms and have them explore for a period of time, 5 minutes or so. Then after a short interval, they're placed back into the Y maze, but now this arm is no longer blocked, so they can explore this arm. But of course, this arm, compared to the other arms, is novel. And what we find is that his performance of control animals, and what you can see is that during the novelty preference test, they show a preference for novelty. So they spend a lot of time in the novel arm compared to the familiar arm. And hippocampal lesion mice, unsurprisingly, don't show this effect. So they don't show a preference for the novel arm. Our control animals from the experiment looking at genetically modified mice, they also show this novelty preference. But our mice that lack the GLUA1 subunit of the AMPA receptor don't show this effect. So what these results tell us is that spatial reference memory, like the performance in the Morris-Wharton maze, is hippocampus dependent, and also performance on spatial working memory tasks is also hippocampus dependent, but we can dissociate them with this manipulation of the GLUA1 subunit. So only spatial working memory depends on GLUA1, and spatial reference memory doesn't. So it seems that the hippocampus is playing a role in both of these processes, but you just wouldn't be able to see that dissociation with damage to the hippocampus alone. You have to have a more subtle manipulation to be able to differentiate between those different processes. Okay, so that's spatial learning in the hippocampus, and of course spatial learning is a property of episodic memory, being able to remember the where component of an episode. But the hippocampus is also important for timing, so the when component of episodic memory. So there's various different memory methods for studying memory for temporal intervals or timing ability in animals. But a procedure that I've been using recently is the temporal bisection procedure. So I'll show this. I'll describe the procedure that we would use in humans, and then I'll tell you the kind of thing that we would do in animals to be able to study as the same process. So in humans, people would be sat down in front of a computer, and they would have to learn a discrimination between a cue that varies in duration. So they're told that a stimulus will appear on a computer screen, and it's usually something not particularly exciting, such as a coloured dot, but it may come on for a short interval. And what participants are required to do is to make a particular response, say press a particular key when it's a short trial, and that will be correct. But if they make a different response, that will be incorrect. So for example, pressing the Z key might be correct when it's a short trial, but pressing the M key will be incorrect. However, when the same stimulus is presented for a longer period of time, the response contingencies are reversed. So now they have to make response 2, pressing the M key, and not make response 1, pressing the Z key. And people find this very simple, and they learn it over a few trials. But what we can do to be able to assess their perception of the passage of time is that once they've acquired it, we can present probe trials of intermediate durations. So a little bit longer than the short durations and a little bit shorter than the long duration, and we can ask people how they what they think it is. Do they think it's a short trial or a long trial? Do they think it's a short queue or a long queue? And what you find is that that when it's relatively short, people think it's short, but somewhere in the middle, they become confused and can't tell whether it's a short or long queue. So they press 50-50 on the short and long queue. So it gives us this, what we refer to as the temporal bisection point, the point which they perceive that duration as being equally similar to the short duration and the long duration. And it gives us a measure of where people think the time has changed from being short to being long. So basically it's a temporal categorization task. Now in rodents, we do the same thing, but they're pressing levers to get food reward. I'm sorry for my very poor quality drawing, but here's a mouse that has an option to press one lever, the lever on the left or the right lever, and sometimes pressing on those levers will lead to getting food that's presented in the food rail. So what happens here is that the mouse learns to press the levers, going between the two levers, in order to maximize chances of getting food. With mice, we take this, I use this particular procedure. So on a trial, the levers are inserted into the box, and on 50% of trials, there will be what we refer to as a short trial, and on the other 50%, there'll be a long trial, and the mouse doesn't know which trial it is. So importantly, when it's a short trial, pressing on one of the levers, so either the left or right one, whichever one's been allocated as being the short duration lever, pressing on that lever after a short duration will lead to food. So that's when the trial ends and the animal gets some food. But pressing on the long lever will never lead to food. So the animal has to learn to initially press on the short lever. On the other 50% of trials, the long duration trials, now pressing on the short lever won't lead to food, and the animal has to press on the long lever, and which happens after a long duration. So what should happen here is that the animals over the course of training should learn to initially go and press on the lever that pays out after a short duration. But if it doesn't pay out, as in deliver some food, they now need to shift their response to the long lever. And so theoretically, what we hope is that performance will look a bit like this. So if we plot the proportion of making the long lever presses over time, over the long duration, they should shift from making mostly short duration responses to making long responses. And of course, the duration at which they're making equally short and long lever presses is our bisection point, and the point at which they can't differentiate between those two durations. So that is exactly what performance looks like. So here are the results of an experiment where mice were trained with two levers. One was reinforced after 8 seconds and the other lever was reinforced after 64 seconds. And here, over the course of the long duration trials, we can see what happens. So they initially make short lever presses. So the proportion of long lever presses is 0. But round about 16 seconds, they shift to making long lever presses. And you can see that the shift from making short lever presses to mostly making long lever presses happens over a space of a few seconds. So it's a very rapid change in performance. So you can get various measures from performance on this task, but the key one we're interested in is the point at which the bisection point occurs, the point at which they can't tell the difference between the short and long duration. So the reason why we did this is because we wanted to look at the function of the dorsal hippocampus. So in this particular experiment, animals were trained up. So here they are here, trained up, and then we inactivated the dorsal hippocampus using DREADS. So this is designer receptors exclusively activated by designer drugs. So using a viral vector, we have the dorsal hippocampus ends up expressing a particular receptor type which is sensitive to the drug that we use. So in this case, it was clozapine. So here, when animals were given clozapine and we were able to inactivate the dorsal hippocampus, what we found was that the bisection point became shorter. So actually, they started to bisect about 2 seconds before training, whereas our control animals didn't show a particular change. So what's interesting about this is it shows that all other measures of performance were very similar across mice. So inactivating the dorsal hippocampus didn't change sensitivity to the passage of time. It just changed where the change happened. So we can say that the dorsal hippocampus seems to regulate how an animal experiences the passage of time. But given various theories of how animals do timing, it's consistent with the idea that the hippocampus controls the rate of like an internal clock. So without the dorsal hippocampus, it's as if your internal clock is running faster, such that you think that things feel longer, quicker, if that makes sense. So these results show that the hippocampus, we're able to work out its role in spatial learning, and also in its role in timing. So various properties that you may associate with episodic memory. So moving on to a different subject, but related, it's all learning and memory. So we know that various brain areas play particular roles in how you represent the associations between actions and particular outcomes. So often when we think of characterizations of why an animal may press a lever for food reward, we might refer to it as a stimulus response learning, because that is literally what the animal is doing. It's presented with a stimulus and makes a particular response. But actually, the animal may not be just forming a stimulus response association. It may be doing something more complex. So an old idea, and when I say older idea, I'm thinking of people like Thorndike, suggested that animals learned simply and only by forming stimulus response associations. So if a stimulus and a response occurred in the presence of something that was reinforcing, such as food, then the association would form between a stimulus and a response. So for example, if there's a lever and you press a lever and you get food, then a lever press association is formed, and the animal is then more likely to press the lever when it experiences it again. Importantly, with a stimulus response association, it doesn't have to know that the response will lead to the outcome. The outcome is just the glue that allows these two things to become associated, but it will just do it blindly without any knowledge of why the response leads to anything. So yes, an early characterization of what learning in animals may be is simply that it's a stimulus response association with no knowledge of what the response really leads to. However, it could be that actually that the stimulus becomes associated with the response, and the response is associated with the outcome. So the animal may press a lever because it has some expectation that it will actually lead to a particular outcome. So it's doing something slightly more complicated, and maybe we would say more intelligent, than simply learning about stimulus response associations. And what we know from the study of learning in animals that there is a myriad of associations that can form between these things. So actually there might be reciprocal associations between the stimulus response and the outcome. One of the people who pioneered the research into what we refer to as goal-directed learning, so learning about the association between a response and outcome, was Tony Dickinson from Cambridge University. And here's some work that he did in the early 80s. So to be able to show that animals learn about the consequences of their behavior, he did this particular experiment. So he had different behaviors, pressing one lever versus pressing a different lever, and he had them lead to different outcomes. So lever 1 would lead to some sucrose solution, whereas lever 2 would lead to some grain pellets. So the animal may if the animal learns an association between its response and the outcome, but it will learn that these levers lead to different things. But in order to be able to demonstrate behaviorally that the animal has some expectation of what it leads to, what Tony did is that in a second stage, after they'd learned these behaviors, he changed the value of a particular outcome. So we call this the revaluation or the devaluation stage. So in this particular experiment, some animals were allowed to consume sucrose, so the outcome that was paired with lever 1 in the training stage. They were able to consume sucrose, and then they were then they acquired a taste aversion to it. So they gave the animals injections of lithium chloride, which leads to the animal developing a taste aversion to sucrose. For other animals, they were allowed to consume pellets, and then they were given lithium chloride. So if animals have a taste aversion to sucrose, behave in a goal-directed manner, what we would expect is that in the test phase, when they're presented with the levers, they're less likely to press lever 1, which led to the devalued sucrose, than lever 2, which of course leads to pellets, which for those animals wasn't devalued. And of course, vice versa for the animals that had pellets being paired with lithium chloride. So here's what Tony found was that indeed that if sucrose had been devalued in the second stage, you stopped pressing the lever for sucrose, but you were carried on as normal for the lever that was paired with pellets, and vice versa for animals that had pellets that devalued. However, what Adams and Dickinson found was that this was only true if animals had a limited amount of training with these two behaviors. If they had a lot of training, so here extended training with the two levers, and then you did the devaluation stage, you didn't see any change in performance. So what they suggested was that initially behavior is goal directed, as in the animal learns to press the lever because it has some expectation that the lever will lead to a particular outcome. However, if you do this over and over again, it becomes habitual, as in the outcome is no longer important, and actually your behavior becomes governed by simple stimulus response associations. And of course, we'll be able to relate to that. There's various things that you do repeatedly, and then sometimes you do it in circumstances where it's inappropriate to do it. You're just doing it out of habit rather than the expectation of what it leads to. So this has led to a lot of work on a network of brain areas and their involvement in goal-directed and habitual behavior. So here, the striatum is very important in receiving input from lots of these different brain areas. So it receives input from the sensory and motor cortices, but also from the medial prefrontal cortex, which we heard about earlier and being important for executive function, receives input from the amygdala, etc. So it seems to be an important network for integrating information that leads to behavioral responding. So I'll tell you about a couple of studies that have used this outcome evaluation procedure to be able to dissociate different forms of learning. So here's a study by Bernard Blaine looking at the role of the basolateral amygdala. And he trained rats up to be able to make different responses. So here, lever pressing versus pulling a chain. So a chain is hung from the ceiling of a box, and the animals pull it for a particular outcome. Then one of the outcomes associated with one of those behaviors was devalued, and animals were then given a test where they were able to make the two responses freely, and he looked to see whether animals were behaving in a goal-directed manner. So what we can see from the control animals is that they respond lots to make the response that wasn't associated with a devalued outcome, so it was associated with a valued outcome, but they made very few responses for the response that now leads to the devalued outcome. Whereas animals that have damage to the basolateral amygdala didn't show this devaluation effect. They made equal responses on those two manipulanda. So the basolateral amygdala seems to be important for goal-directed behavior, and actually the story gets more complicated because there's brain areas seem to have particular functions in that may impact in goal-directed behavior. And so in terms of the BLA, the general idea is that it seems to be important in representing the sensory properties of the outcome. So here's another study that's used the outcome evaluation procedure done by Simon Kilcross and Etienne Coutreau some years ago. And they were looking at the medial prefrontal cortex. So the medial prefrontal cortex has the infralimbic area and the prelimbic area, and they seem to have dissociable roles in goal-directed behavior. So in this study, they used the outcome evaluation test to look at the functions of the infralimbic and prelimbic regions. But for some animals, they had a low amount of training, which of course we would associate with being goal-directed. And then for other animals, they had a high amount of training, which should lead to habitual behavior. And this is what they found. So when animals had a low amount of training, so they should be acting in a goal-directed manner, control animals showed sensitivity to the outcomes evaluation effect. They made very few responses for the response that had been led to the devalued outcome. Lesions of the infralimbic area were without effects. They didn't affect these animals were still goal-directed, but damage to the prelimbic area impaired performance on the test. So they were now no longer acting in a goal-directed manner, suggesting that the prelimbic region is important for goal-directed behavior. When animals had a high amount of training, these sham animals, just as you'd expect, were no longer sensitive to outcome devaluation. And unsurprisingly, animals that had damage to the prelimbic region were also insensitive. But interestingly, the animals that had damage to the infralimbic area were still goal-directed. So the infralimbic region plays a role in becoming habitual. So if you have damage to the infralimbic region, it maintains goal-directed performance. So this suggests that actually there's interaction between the stimulus response learning systems and the response outcome learning systems, and that they compete for performance, and different brain areas are regulating which one is expressed. So we can think of it a bit like this. So initially, the prelimbic region is important for response outcome associations that seem to govern behavior when you've had a limited amount of training. But when you've had more training, what happens is that you end up being habitual, and this seems to be because the infralimbic region actually inhibits goal-directed behavior. It stops the expression of goal-directed behavior, and we know that if you train animals up to become habitual and then you inactivate the infralimbic region, they switch back to being goal-directed. So it's not that animals are initially goal-directed and then become habitual, and it's just a sequential progression. The two things are happening in parallel, and all you need to do is to turn one off for animals to switch back to being the other system. So these associations between brain areas tells us about the underlying psychological processes. So my last example of interesting ways of studying behavior, hopefully I'll be able to do in the next few minutes, but it's a nice one to end on. So this is some work that I've done looking at feeding behavior in rodents in relation to learning. And feeding behavior is very interesting because it allows us to dissociate different properties of what the animal is learning about. So when a rodent licks a zipper tube for a particular substance, it does it in a very particular manner. So the rodents, so rats and mice and certainly other rodents, make clusters of licks. So that they'll make a succession of licks in very quick duration, and then there'll be a pause. So they might go lick, 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 then a bit of a pause, and then another lick cluster, and so on. But what we know is that different factors affect how much a rodent drinks on any particular occasion, but then other properties of a substance influence the size of these lick clusters, and the amount you drink and the size of a lick cluster seem to be independent. You can dissociate them to the two. They tell you about different factors that are affecting behavior. So here's some work that I did some years ago, and here mice are just consuming different concentrations of sucrose, and I was looking at the effects of deletion of the GLUE1 subunit. So these are the mice that I told you about earlier that have a deficit in spatial working memory. But for various reasons, we wanted to look at the role of the GLUE1 subunit in feeding behavior. And what you can see here is that both groups, the control animals, the wild type mice and the knockout mice, they consume similar amounts of the different concentrations of sucrose. But importantly, the amount of sucrose that animals drink changes as a function of sucrose concentration. So if it's 4%, they drink quite a lot, but if it's 32%, they drink less. And what we presume here is that as the concentration increases, they become sated. They like 16% sucrose, and I'll show you measures that suggest that, but they can't drink a lot of it, and similarly for 32%. When you look at the size of lick clusters that the mice make for the different concentrations, what you find is that the lick cluster size goes up with sucrose concentration. So they make larger lick clusters, more licks per cluster, for 32% than for 16 and 8 and so on. So people have suggested that lick cluster size gives you a measure of palatability, how nice the particular substance is. So it gives you a measure of the hedonic reaction to the substance. And what we can see is that although our GLUE1 knockout mice consumed similar amounts of sucrose, when they did, they consumed it in much smaller lick clusters. So this suggests that GLUE1 may be important for the hedonic reaction to sucrose. If that's the case, then we may think that this may impact on the animal's ability to learn about sucrose. Well, that doesn't seem to be the case. So here is a simple flavor learning procedure. So in this procedure, animals consume different flavors on different sessions. So they have sessions where flavor A might be paired with a high level of sucrose, so 32%, and flavor B is paired with a low concentration of sucrose. And then in the test phase, both flavors are paired with the same level of sucrose. And what you find is that control mice and knockout mice show a preference for drinking flavor A over flavor B. And this reflects learning because, of course, the concentrations of sucrose in the test phase are the same, so the performance must depend on what they learned from the previous training situation. They like flavor A more than flavor B because it was paired with a higher concentration. So even though GLUE1 seems to be important for the palatability response, it doesn't impair their ability to learn about sucrose. So this leads to the problem of what does the role of palatability play in learning about sucrose? Well, it could be that for GLUE1 knockout mice that they are insensitive to its hedonic properties, or perhaps they are sensitive, but they just can't show it behaviorally. They have impaired lick cluster sizes, but it doesn't mean that they don't know about the hedonic properties of sucrose. Well, there's a way of which we can dissociate those different accounts. And what we know is that recent experience of sucrose changes how you feel about sucrose. So in this procedure, we know that if you're allowed to drink 4% sucrose, but if it's preceded by different concentrations of sucrose, then it changes how you feel about 4% sucrose. So if you drink 4% sucrose, but it was preceded by 1% sucrose, you make large lick clusters for 4%. You drink it as if it seems to taste nice, high palatability. However, if you drink 16% sucrose before drinking 4%, you then make very small lick cluster sizes for 4% sucrose. You drink it as if it's less palatable. So we can manipulate seemingly the palatability of 4% sucrose by what you've experienced recently. So we use that procedure to be able to affect how you learn about different flavors being paired with 4% sucrose. So in this procedure, animals were allowed to consume different flavors mixed with 4%, but one flavor was always preceded by 1% sucrose, and another flavor was always preceded by 16% sucrose. And then in the test phase, you were just allowed to drink flavor A or flavor B paired with 4% sucrose. So if this prior exposure to sucrose had affected how you feel about 4% sucrose, it should determine how much you drink in the test phase. And what we found is that it had a very similar effect for both control mice and for GLUA1 knockout mice. So although they show impaired lick clusters, suggesting they have impaired palatability, they do show normal sensitivity to changes in the effective palatability of sucrose from recent experience. So it suggests that really they have a problem expressing hedonic reactions rather than actually a problem with anhedonia generally. So there's an example of things we've learned from various experimental procedures that we've used to dissociate different accounts. So that's the end of my talk. I'm going to end with a quick plug for Durham. So I'm in the Department of Psychology, and we have a large group of people all doing work on learning and memory in rodents with various using various different neural manipulations. So, for example, we have a bunch of people doing electrophysiology, recording neurons in the hippocampus and other brain regions. Jasmine Strickland is using neuropixels to measure large areas and the firing rates of a large amount of neurons involved in various forms of learning. And we have people looking at hippocampal function generally. So I won't dwell on that too much because I know we're running out of time. Thank you very much.
Excellent. Thanks, David, for a great talk. I found the last example you mentioned about the flavor quite interesting. I want to ask a couple of questions on that. But first, we have a question from the participants. Sorry if I missed this. What model of disease or damage to the hippocampus is being explored in humans by the use of the animal behavioral test mentioned? So I guess in the first part of the talk.
Well, lots of those studies were done just to look at the function of the hippocampus generally, but we know that in terms of Alzheimer's disease, the first place you'll find degeneration is in the hippocampus. Mm-hmm. And certainly when people look at people here in Durham are looking in humans to have find procedures that you could give to people with mild cognitive impairment to test whether they would develop into full-blown Alzheimer's disease. So basically, when people go to their doctor and say they're suffering from memory impairments, but you probably miss the opportunity to have some kind of intervention. So people want to have better ways of being able to look at hippocampal function to be able to spot these deficits early. And so Anthony McGregor and Colin Lever in the department are using various methods to do this, using spatial tasks that you can do on a computer with people to be able to look at early predictors of neurodegeneration.
Yeah, excellent. I guess this is a bit related. Maybe it already answers this one. So very insightful. Thank you, Professor. I'm a little bit curious about what neuropsychiatric or neurodegenerative disorders you work on using all the behavioral tests you mentioned.
Well, so it depends on the particular questions, but I'm interested in various kind of the role of learning and memory in different neuropsychiatric disorders. So I didn't actually talk about this work but today because some of the procedures get a bit complex. But Marta referred to dopamine and its role in cognition. And what we know is that in animals that it seems to be very important for what we refer to as prediction error. So that animals and people learn by making predictions about what they think is going to occur in the environment, and whether that's borne out then impacts your learning. So if there's an error in your prediction, you update your learning. And dopamine has a very important role in those processes. And of course, dopamine, as well as Parkinson's, is also associated with schizophrenia. And people have suggested that some of the symptoms of schizophrenia are related to impairments in prediction error. Yeah, so some of the work I've been doing with the glutamate receptor knockout mice are related to that kind of question.
Mhm. I had more of a general question in terms of on the technical side. So how reproducible are usually these behavioral outcomes across different labs and mouse strains?
Uh, uh, well, so it depends on the various tasks. Lots of these procedures are very reproducible. So the Morris-Wharton maze works over various different labs. What we know is that you have to be very careful about the spatial cues. So one of the inherent difficulties with spatial tasks is that people will use different cues, and they may be so you can't reproduce those spatial cues entirely across labs. But as long as there are sufficient allocentric cues for an animal to use, you see those kind of effects. If you're doing stuff in automated apparatus, like in a Skinner box, an operant box, then you can reproduce the procedure and the results very reliably across labs. With stuff like T-maze alternation, it's a very simple procedure which animals show in all sorts of circumstances, so it's very reproducible. There are work looking at spontaneous exploration behavior, say with like objects, and I think that is more prone to variation depending on the objects you use and the environment in which you do it. So if you're looking at spontaneous behavior, the worst thing that could happen is that you put an animal into an arena where you want it to explore and learn, and it doesn't do any exploration, maybe because it's anxious or whatever the factors are. So those things can be harder to people do them all sort over or in different labs across the world, but there is variation in that, and depending on the procedures you use.
Yeah, great. Okay, there's another one. You mentioned spatial memory and the temporal bisection task. Does GLUA1 play a role in brain process involving time, space, or both?
That's a very good question, which I can't completely answer. So I haven't tested the GLUA1 knockout mice on the temporal bisection procedure, but what we know is that it's important for sensitivity to rate information. So in a normal kind of Pavlovian conditioning experiment where, say, a light leads to food, the duration of the cues is critical in determining the amount of responding that an animal shows, the amount of learning. And we know that GLUA1 seems to render animal, if you knock it out, it seems to render animals insensitive to that rate information that's provided. Now, whether this is because it plays a role in spatial learning and timing, and therefore these are all related to episodic memory, or whether actually it has separate functions within spatial memory or temporal timing, we don't know yet, but it's an interesting question.
Okay, I wanted to ask a question on the last example you mentioned. So with this flavor exercise, you mentioned the fact that the animals were able to kind of change the expectation when you had one drink and then the other afterwards. What was the gap between both drinks? So how long does this effect last?
There it would have been a number of minutes. I can't remember exactly, but it might have been something like 10 minutes. We haven't tested how long it would last for, but we presume that it would be that it would go away with time. So exactly what's happening there, we're not sure, because there are various things that could go on. So people refer to successive negative contrast, where the animal is able to actually compare at some level the concentrations it's had, such that it deems one of them negatively. It deems it less palatable because of the prior experience, and yet that's a kind of psychological characterization of what's happening, but it could just be that it's becoming adapted to there's some kind of level of sensory adaptation. So maybe if you drink 16% sucrose, you have sensory adaptation to sucrose such that 4% doesn't taste as nice. And so that effect should last for however long sensory adaptation lasts for. You see those kind of negative contrast effects over durations which are longer than you'd expect for just purely sensory adaptation, suggesting that the animal is learning something about the properties and has some kind of seemingly like emotional reaction to being presented with a particular concentration. It's as if the animal is disappointed. I was expecting 16%, but now I got 4%. Therefore, this 4% now tastes worse than it would have been otherwise.
So I guess we'll take one last question. We're running a bit over time. There's one from N. Jordan. Did you see or know of research looking at this where, for instance, long-term impairment might be seen in different diets, high UPF versus no UPF?
In different diets?
Yeah.
So I haven't done any work on that, but there are studies looking at how diet affects cognition. Is that what the question was asking?
Yeah, I guess it was in terms of the flavor learning. Yeah, whether I guess it, and maybe can clarify, but my reading is that, it's related with the flavor learning where this is related with diet, with ultra processed foods, but I guess this is not what you really tested in this case.
Yeah, well, I don't know about flavor learning specifically, but people have looked at the consequences of having a high fat diet and leading to cognitive impairments, and leading to cognitive impairments independent of learning about food. So in these kind of spontaneous learning paradigms where animals are allowed to they explore objects, for example, and you test their memory for the objects, a high fat diet leads to performance which is similar to having hippocampal damage. Yeah.
Do you see, I mean, maybe this is a bit of a naive question, but do you see any potential applications of this flavor learning for weight loss practices, whether people can potentially, associate a certain flavor with high sugar, but then try to reduce the sugar and still keep sort of the pleasurable from the food? Yeah, I assume you haven't really tested this, but I wanted to get your thoughts on this.
Yes, yeah. So, so we know that learning and memory will impact how much you eat and then how much you think about it. So, how much you value it or how you feel about it emotionally. And people have looked at various manipulations to kind of see how it impacts feeding behavior. So one of the reasons I got into this was I was interested in kind of like short-term habituation of effects and how that affects how you learn and also what you learn. Suzanne Higgs in Birmingham University does a lot of work in humans based on basically expectations of what you're going to eat versus what you actually eat and how that impacts it, how much you eat. So can control the extent of feeding behavior based on your expectations of what you're eating. Yes, it's an interesting area whether you can actually have some real intervention which you could apply to the public. I'm not sure, but interesting question.
Very interesting. Thanks so much, David. Thank you for both speakers, Marta and David, for this excellent session. So we'll be back tomorrow for our last Essential GIBA Concepts and Techniques session on immunology, being presented by Joanna and Fran. So we'll be back tomorrow. Again, as I mentioned, the recordings will be shared at a later point. I'm sure Marta and David will be happy to get any further questions by email if people are interested as well. So thanks everyone for joining. Thanks again, Marta and David, and see you all soon.